So hi everyone, for those I haven't discussed yet with. So today and tomorrow I will try to talk about marine ecology and especially Antarctic sea ice associated ecosystems. So I know you all come back from a physical oceanography or glaciology or paleology background, and so it will be very basic today. And tomorrow will be more about marine, Southern Ocean marine ecosystem assessment, about what is the state of these different ecosystem compartment and how, and what are kind of the projection as well for these compartments. But briefly first, today I will talk about something very general, but usually I work on seals, mostly the ecology of seals, which marine ecology, it means studying the species in their environment and understanding how the physical habitat and the biogeochemical cycle will influence the ecosystem and then up to these predators. And the idea is to understand how they live and how their life history traits, so like their breeding success, their survival, their resting times, how is it influenced by the environment, and how these top predators can give us information on underlying trophic levels and then the food chain in general. And so for that we use biotelemetry to study this feeding behavior, breeding, resting, molting, survival. And we have like continuous diving profiles of animals that we study. And based on that and the physical habitat that we can study from these loggers, we make inference about where do they feed and where do they go and how does it change in time and space and why. But today I'm not going to talk about that, so it was just like a brief introduction of where on what I work. I really enjoyed David Marshall's presentation, and I think I copy like a different. I'm an early career scientist. I just got a position 2 years ago, but I think it's important to show you my small path, I would say. And I had an unusual path because I spent like 16 years in a pool doing artistic swimming. And actually, I had this kind of random behavior of the seals, like random walk trajectories. And I was like a nice fish thinking what I am doing in a pool for like so many years, like why? At the end, I finally ended up to the Olympic Games, and that was great and great experience. But I was like, I am so bad at holding my breath. Why I not all study the best diving animals? So Southern elephant seals. So no correlation with that, actually. But actually, I had the chance to work during my master on Southern elephant seals, and then I started a PhD on Southern elephant seals, but only when those guys go to Antarctica, and I was trying to understand why, from subantarctic island, why do they go in a straight line to Antarctica to feed and eat and come back to the colony? So I spent my whole time trying to make some link between their diving behavior and sea ice, and work a lot on coastal polynya as well, if you have questions about coastal polynya. And I did the PhD between l'Océan and Tasmania, so the baguette and the Tasmanian devils. It was great, but then I found these guys were really smelly and ugly, and I was like, oh, maybe I should go towards these like super pretty emperor penguins that are nice and always clean and but it was still in sea ice, so I was very excited, like great, another diving predators, but now it's a bird. So I did my postdoc in Woods Hole Oceanographic Institution. I stayed there almost 3 years. I saw on the slide of Sara that I spent a little bit of time at NCAR and really enjoyed this place. We were working on projection of emperor penguin, and we had a project with Marika Holland over there, and this place is incredible. I was really happy to discover this place. But then, this nasty penguin here, penguins are not really collaborative in their research. I found the penguin community quite not nice, and I was like, and I started to think maybe I will quit science, because for me it's not enough collaborative, it's too much competitive, and I already did too much competition, and I don't want to hear about that. And someone that is here and someone else convinced me to go back to these sausages and say, come back to France and apply to CNRS, stop doing penguins. And I applied to CNRS, and I was very lucky to get a permanent position 2 years and a half ago. And I went back to l'Océan and the baguette. Really happy after 3 years in US. And a glass of wine. And here I am. So basically, I am working on marine ecology, and the idea is to understand the behavior of marine predators underneath the sea ice, and I'm in the frontier between physical and oceanography, glaciology, and biology, physiology. So I'm a little bit doing split all the time and trying to understand the different compartments. So if I said anything wrong, please correct me, because I am struggling to have the big view. Yeah, okay. So now we start. So something I wanted to show you before we even start on marine ecology is the Global Assessment Report on Biodiversity and Ecosystem Services, and it is just a small paragraph from the status and train. And I think anyone involved in biology should read at least the summary for policymakers, or at least the whole report. But human actions threaten more species with global extinction now than ever before, and 1 million animal and plant species already face extinction. And this is, for me, the most frightening thing is like the rate of species extinction is already at least 10 to 100 of times higher than it has averaged over the past 10 million years. So I think the goal of marine ecologists, maybe some of them, is to try to understand the drivers and all the anthropogenic pressures, including climate change as well, but on these species, and trying to explain to people, the general public, what is happening, and like, are the population are stable? That would be great. Are they adapt to their changing environment? Maybe yes, maybe not. Are they actually not adapting and struggling? And what are the different pressure? And try to push for reducing our carbon footprint that will only make a change. And I want you to think about one thing. It's like, do you want to live in a place, in a world where we have only humans and non-humans, living things are not there anymore? Because sometimes people ask me, but why we want to save penguins or seals or krill or theropods? And I'm like, yeah, what is the meaning of living in a world with non-human living species? Yeah, but it's an open question. Maybe some people will enjoy not having any other mosquitoes around them. Okay, sea ice. So sea ice is a major structure component in the Southern Ocean that will affect Antarctic ecosystem in many ways. So here it's the satellite images that describe the different type of sea ice, and I think Martin and Dirk will maybe talk about that more. But basically there is the marginal ice zone, the pack ice, and the fast ice, and all will have different properties, and that will change the way the species live within, inside, at the bottom, on the top of it. And it's about lots of different species and ecosystems that will be really adapted to the phenology of the ice. And when I talk about phenology, I always think that ice is like a braised lungs. I don't know how to say that, like a lungs braising. And maybe it's braising less and less, but it goes from like 3 million kilometers square to 18 million kilometers square in a season. And so really like lots of species, primary production, phytoplankton are really adapted to this seasonal cycle, and it's one of the major seasonal physical change in the ocean that happens every year. So first, it's a very basic scheme coming from the Meso papers that I will talk about tomorrow. But you have the fast ice, the pack ice, and you have different communities associated with it, and I will go a lot in details through the talk. But basically, we have first communities of fish living just underneath of the sea ice in different type of sea ice. We have also lots of animals living in this Antarctic brine channel system that are very cold and salty. And you have lots of species of calanoid copepod, arpacticoid copepod, also pteropods, and you have like thousands of individual per square meters of ice. We can also have like high concentration of krill, ice krill, because there is Antarctic krill and ice krill, and I will come to that soon. So ice krill is associated with ice shelf and polynya, and we have also salps that is going a little bit in competition for food resource with krill. So salp will mostly develop in low chlorophyll, warm waters area, while like krill will develop in usually high chlorophyll and colder water area. And then within the phytoplankton, we have like pack ice communities, diatoms, and we have also surface community dinoflagellates, and we have also lots of algae associated to the ice. So this is just a basic overview. So today I will split my talk between pelagic assemblage, which I mean like everything that is from the slope offshore within the Antarctic area, and Antarctic shelf assemblage, of course, everything that is on the shelf. And I will make just an emphasis on krill and fast ice communities. And tomorrow I will more talk about marine ecosystem assessment for the South Ocean. So the pelagic assemblage, I like simple scheme, but basically there is different way that the energy goes through different pathway. Yeah. Communities, a group of species, a group of a community of different species living together in the same habitat, I would say. Yeah. So you have different pathway, and so from phytoplankton you can go through Antarctic krill all the way to predators, or either you can use the other way. So the Antarctic krill is the major pathway, but it's been new studies, I mean not new, but like recently it's been still recent. A second major pathway that has been evidenced in the pack ice, mainly in autumn and winter, that goes through copepods and fish and predators. And it's important to understand the different pathway, because if something happened here and you don't know the other pathways also existing, like you can't make inference about the future of ecosystem. So through this pathway, I mean, mesopelagic ichthyophauna, fauna, which means fish, sorry, is composed of 73 species, and we have dominance of myctophiids in biomass and in terms of species. And south of the polar front, it's mostly the myctophiid Electrona antarctica that is there. And Electrona antarctica is very interesting because it has like a dual vertical migration as many species, but very efficiently transfer energy between the different compartments, epipelagic and mesopelagics. But about 24 to 70% of the biomass of Electrona antarctica is from 0 to 1,000 meters. But what is nice is that they found in the upper 200 meters at night in the Antarctic pelagic habitat, and play an important role as they are very abundant in biomass to transfer the energy. So Electrona antarctica may represent an alternative pathway as well in the pelagic food web. So going from Antarctic krill to here, so it's all these dotted arrows, and also through copepods. So very important component as well. So what I said already is that they inhabit deeper waters, but in pack ice area, certain species can be found just underneath the sea ice. So for years, people were thinking that they were only like moving through the water column and being in deeper waters. But finally, they found under the pack ice this aggregation of different fish that were not usually expected there. So different example. So krill. Krill is one of the key components of the Antarctic ecosystem, and I will go back again on that. But there are lots of uncertainty about the future of krill, because part of their life cycle is associated to the sea ice habitat. So the larvae will hide and feed within the sea ice. So for years, people were thinking that they can't live without sea ice. There have been like 1,000 papers on that. And then now they are questioning more and more whether they can live or not with sea ice. And there are lots of uncertainty of the distribution of adults within the sea ice and also the role of sea ice for the larval development. So lots of questions that is really problematic to understand, given the sea ice change we are experiencing, what is the consequence on the general food chain. So I have 3 slides about the krill cycle just to explain you what is expected in theory, but everything of that is put into the question now by different papers. But basically, the cycle is they spawn in spring, go at depths, go back, and the larvae will hide under the pack ice, while the adult, which I call krill post larvae, will be underneath the sea ice in spring. I mean, the melting sea ice, feeding. Then in the winter, autumn, there is a segregation between the adult and the larvae, and it's to prevent cannibalism, which happen a lot. So if you don't have sea ice, the adult will eat the larvae, which it's the first question about the role of sea ice for krill. And then in winter, we don't know. We think that they move night and day, but we don't know actually the real distribution of the adult within the sea ice. And the larvae remain deep in the sea ice. This is the pelagic community, which means on the slope and offshore. It's not on the shelf. Yeah. 1 or 2 years. Year 3, they become adult. Okay, and so this paper was like atomic bomb. Maybe not, but for me, yes. Bettina Meyer and her team show for the first time that actually sea ice was not really a feeding habitat for larval krill, but was more like a shelter habitat to protect from predators and stuff. That actually krill could live, the larval could survive with almost no food. And it was super interesting. They succeed to do that by making they have divers that have been there and like moving and trying to understand how the larval krill were distributed. And this is super hard because the larval krill always avoid anything, and the krill as well. So it's very hard to study. They're just like little shrimps that will just avoid you all the time. So when you sample them, it's very difficult as well. But basically, they found them mostly on this terrace, and it's also to show you the structure of the sea ice that is not a flat thing. It's all cracks and terrace and cavities. They found they were mostly here and not feeding, just protecting from predators. And they also found something really nice in this paper. It's actually an inverse diel migration, because they found them during the night at depths, the larval krill, and coming up underneath the sea ice during the day, which we didn't know that the larval krill were having this like migration type of style. And also there were like lots of CF drift and ocean current having an effect on this larval krill. So continuing on krill, and you will see where I get, because then maybe you get lost now, but there is all this. So this is the Kamala areas and all the hatch area where there is possibility to fish krill. For now, we are okay in this area. They don't fish too much because it's far away from continent, and there is still sea ice, so it's complex. But in this area, they fish a lot. And of course, they fish, and then they said, maybe there is an impact on the ecosystem, so we're going to make a survey. And then, maybe we should estimate the biomass of krill before fishing or giving stock. And so now they're at the point where they try to estimate the biomass of krill, which would be interesting. They have been estimated already, but they try to really estimate the biomass of krill and also try to understand the consequence of overlap with different krill predators and the fishery. And the estimated biomass from the latest, I would say, literature is 379 million tons. But this is an estimated biomass, and this is very difficult to estimate biomass of krill, because you sample the krill here, and then you move here with your boat, but actually the krill moved as well, and so maybe you sample the same swarm. because the swarm of krill could be several kilometers wide and like could go up to 10 meters deep. So it's huge. And so making this survey, you never know if you sample the same krill swarm that have been moving or not. So you have the chance to overestimate, or you have the chance to underestimate. In case you sample here and then you go here, but you didn't find the patch and you underestimate, because it's a very patchy resource. And the main problem as well that they have to understand the biomass and also to have an idea of where they should fish, whether where they should not, and to prevent the problem on the ecosystem, I would say, is that krill will follow a lot the dispersal, and we'll have, I mean, this is some tests on the dispersal of krill, but there will be lots of dispersal and advection of krill through the currents, and that will also affect a lot where we think the stock is. And there is also seasonal migration, so you can see that the adults will be way offshore at certain time of the season, and then we will come back, and so you have lots of change, and you have also this vertical migration. So then it's very difficult to understand where they are and how much they are. And this is actually the catch of krill since the beginning of the history of the krill fishery. You have gaps that is associated to some technical problems, and also maybe change in the ecosystem, and also the dissolution of the URSS. And then we can see that the krill catch in the area 48, which is like the peninsula, keep increasing. And actually we don't fish anymore on East Antarctica for now. And this is a problem. So we have a species that is very difficult to sample, that is a key component of the ecosystem, that is fished a lot, and for which we don't understand fully the behavior, and also which suffer from some impact of some potentially climate change. So this is a study of Atkinson showing like a contraction of the krill distribution and also a reduction from all the data they had. There are people that are arguing about this paper and thinking that there might be some bias, etc., in the methods, but I think it's the best available science that we get, and like maybe until the next time we get more information and more ideas. But basically, in the Commission for the Conservation of the marine fauna and flora of the Antarctic, they try to understand the krill stock hypothesis to be able to give some recommendation of where they should fish and where they should not overlap with predators. And based on that, they try to do a krill stock hypothesis, and like lots of groups, and I will not go into the detail of all the groups, but there are groups trying to develop this kind of hypothesis where they can draw the spawning hotspot, the place of juvenile recruitments, and also the pathway where all the krill is. And you can see how much it's, I mean, I'm not going to go into detail, but how much it's complex to draw the different spots, the different advection, and try to understand where we should not go fishing and where it's important for predator and the ecosystem. So very complex krill question. And I will skip that, yeah. Another thing that I would like to show you is the also complexity of the link within the food chain. So this is a schematic adapted from this paper, Tribal, where they represented the different type of phytoplankton, zooplankton, and then the fish, the predators, and also they added the fishery part and the benthos. And they made some simulation to try to understand if we have more that or less that or more that, what happened to this and this and this. And it's very complex because you always in a model you can't really represent everything and every compartment. But so I will show you different simulation, but basically, and also the limits of this simulation. Basically, they made a simulation where they have more small phytoplankton and less large phytoplankton. So basically, you have less of that and more of that. And what happened all the way here? And basically, they found that it's like, so blue is negative, green is positive, and this is the number of simulation. And they found that the more small or less large will have a huge effect on the krill fishery, and krill-dependent predators. And they did another simulation where they increased the gelatinous zooplankton, so the salp, and they found negative effect on krill-dependent predators. Positive effect on krill-dependent predators and krill. And that was weird because usually we said, okay, if there is more sap, it will have an effect on krill because there is competition between krill and sap for food. And actually, they didn't include the competition between krill and sap in the simulation. And it's how they discuss it that they end up with this positive value because just the factor of competition was not included. Another simulation is increase of baleen whales through the stop of the whaling fishing. And they found that they will have like a negative effect on krill fishery and on krill. But same, they didn't take into account the role potentially of whale of stimulating primary production through their faces that might have like a strong impact. So same, still some limits in this model. It's in this modelization. Finally, if you do a non-controlled krill fishing, they found that it's very bad for like top predators, baleen whales, and also like all the fish and squid, krill, salp, scoby, but actually most of the components actually are affected by that. So using this simulation, we can have an idea of what would be the impact and trying to understand all the drivers at the same time, because all these components are very difficult to study and observe and have data for that. Yeah, and so another thing as well to think when we look at the picture of the ecosystem. So this is the krill availability for penguins or whales. When you try to combine the effects of climate condition, whale recovery, fishery expansion, and you try to see the consequence on krill, for example. So you can see that krill is increasing, okay, and then there is change in sea ice, and then there is the whale recovery, and so the krill is diminishing. But you think that the whale will grow exponentially and everything will be great. But actually there is a new paper, it's not on South Wales, but it's on North Pacific openback whales that show the trend of two populations of North Pacific openback whales from 2002 to 2020. And this paper was super interesting because it showed the full recovery of whales. And then there is like this huge Pacific marine heat waves that change a lot of the resource for this population. And you can see this decrease of the whales. So like this graph is just to show you that the combination of like whale recovery, availability of resource, climate change, different forcing will have different impact, and that you really need to consider all the factors together to understand the big picture. Okay, back to the now shelf community. So you've seen everything about the pelagic, and now we'll switch to the shelf. So the shelf, Sam, we have like seals, algae, phytoplankton, and we can go through the crystal krill, which is the ice krill, up to the predators, or either we can go through copepods, some fish, and other fish. So in late spring, early summer, this pathway is used until depletion. And once it is depleted, we have this summer pathway. And then when we are more in the fall transition and the Antarctic silverfish actually can't find food anymore, they'll start to be cannibalistic, and so it's a way to regulate as well the food web, where we call that wasp webs. So that means that at the middle they will stabilize the equilibrium in ecosystem. So in the shelf community, the dominant fish species from the surface to 900 meters is the nototenid group, the Antarctic silverfish. And there is difference in spatial distribution. The larvae feed on zooplankton in the surface layer of the shelf water. Then the adult will move offshore outside of the shelf, and the mature adult, the juvenile move offshore, and the mature adult then will return to the shelf to spawn. So you have lots of seasonal movement for even one given species. Yeah, that is not really important. Same, the toothfish is a fish that is highly fished as well. And you can see that juvenile are principally found also on the shelf, while adults are also moving to the slopes. And sometimes we found them shallower than expected, so they move back close to the ice to feed probably or under the fast ice in mid-depth. And to finish on the shelf assemblage, the Antarctic benthic communities are characterized by important biomass, biodiversity, and endemism, and have a strong link as well with what happened in the surface. And I will detail that a little bit more. And they have a strong resistance to change in species composition. So for this reason, they are among the most ecological stable communities in the world. Okay, so within the sea ice, there is lots of different communities, and I will now detail that. You can see here some ice corings, chlorophyll data. So this is the mean of chlorophyll in ice corings for different time of the year. And you can see that you have a significant part of activity happening in the ice. And also this activity, some studies, they found that it's mostly close to the bottom of the ice, and mostly in not too thick, but not too thin ice, mostly around 1 meter ice. And Martin could talk to you about that more. And you always think how do this ice algae survive in low light condition? And actually they can turn into being mixotrophic, which means that in absence of life they will become heterotrophic and consume the organic matter under the sea ice that is released by these brine channel systems. The sea ice ecosystem accounts for approximately 1 to 3% of the total annual production in Antarctic waters, which is not a lot, but actually in certain time of year where there is not a lot of primary production anyway, it's super important for the survival of many species. And actually when you consider only the sea ice zone, it's 2 to 24% of the total primary production. And you can see that there is a huge variability in this primary production depending of where you are in Antarctic sea ice. So at the bottom of Antarctic sea ice, in platelet ice, so platelet ice is some thin piece of ice that is formed from the release of really cold ice shelf water, and that will go underneath the sea ice. Internal new ice, pike ice, bottom, and you can see this huge amount of annual production, daily production for the platelet ice, for example. So the Antarctic fast ice community. So you can have virus, bacteria, heterotrophic and autotrophic protists, which is commonly called sea ice algae. And you can see that you can have this community in platelet ice, in bottom ice community, strand community. You can have also in this huge column of brine channel. And you will have difference in the community depending you have non-rafted fast ice or rafted fast ice that will have different impact. The relative contribution of fast ice algal production to overall primary production for the sea ice cover period in Antarctic nearshore ecosystem has been estimated to range between 50 and 65%. So during ice cover period, which means like the winter time. So it's super important for sustaining marine ecosystem and sea ice ecosystem during that time. Also, the regional variability of bottom ice algal production and biomass is largely controlled by the presence and absence of this platelet ice layer. So I already talked a little bit about that, but a little bit more in detail. How this ice algae will remain photosynthetically active under both changing and often varying light condition. And they found some very strong different adaptation or change or way to handle low light condition and still being active during the winter time in the sea ice. So, for example, they showed the ability of Arctic, but they hypothesize that it's the same for Antarctic. So, Arctic bottom ice penate diatom to vertically migrate in response to changing snow condition or light condition within this channels of ice, and they will move vertically depending on the light condition. Another hypothesis is, not hypothesis, but other capacity, I would say, is the pigment composition and expansion of light harvesting complexes. So they will just change in the way the pigment works and function. Another thing is also the change in the structure of photosynthetic units, so decreasing the number and pigment packaging inside the cells. So it's all, I'm not specialized at all in that, but it's all like specialization in a way that the cells are functioning in terms of the pigment. So it's also very important to think that there is a strong coupling between sympagic, so associated with sea ice, pelagic and benthic coupling. And I think like everything that falls from the sea ice is very important as well for the benthic communities. And the brazing of the sea ice, the seasonal brazing of the sea ice brings lots of materials to the benthic community, and it's why it's one of the most biodiverse and rich benthic community that we found in Antarctica. But pelagic grazers, they will utilize ice algae at different critical time of the year, and the fact that this fast ice area will be in very shallow water depths, it will have an efficient export of the ice algae to the seafloor, resulting in a tight cryopelagic benthic coupling with this organism. And also something to keep in mind is like how thick and how much snow and how much the ice is formed will have an influence on the light availability, and also how much persistent is it, like a multi-year ice or just a single layer ice. It will result in a decrease or increase in pelagic and benthic primary production because it will reduce all the phytodetritus and all the organism going to the benthos. So it also have like an importance for marine ecosystem. So here we have the ice shelf, fast ice, and here like fast ice and pack ice. So again, like this platelet layer, you can see the under ice grazer, copepods, diatom, and you will see that mostly small organisms that are tolerant to high salinity, very cold temperature will be found in this brine system within the ice. So basically it's copepods, turbellarians, different species. I mean, you don't need to remind the name, but I just like so you get an idea of which organisms are living within this system. And the bigger zooplankton, so amphipod, ophioziacids, theropods, large copepods, they will mostly live at the sea ice interface. This platelet layer will be super important as like spawning and nursing grounds for many species of fish, especially Antarctic silverfish. And it's very interesting in terms of how much it aggregates some biology around this area. And lots of fish actually will be just nearby ice holes or at the ice interface being there. Yeah. And I think that's all for today on this part. I was a bit speed. But